The Garage Gene Revolution: Editing Life in the Age of DIY Biology. Imagine a world where the power to manipulate the very code of life is no longer confined to sterile laboratories and the hands of elite scientists. What if genetic engineering tools became as accessible as 3D printers, available to anyone with a curious mind and a garage? This isn't science fiction. It's the burgeoning reality of DIY biology, a movement that's rapidly shifting the landscape of genetic research. From university labs to suburban garages, the tools to alter DNA are becoming increasingly democratized. This shift holds transformative potential, promising breakthroughs in medicine, agriculture, and environmental science. But it also carries inherent risks, raising profound ethical questions about who should wield this power and how it should be controlled. The allure of DIY biology lies in its promise of empowerment. Individuals can take control of their health, exploring personalized therapies and preventative measures tailored to their unique genetic makeup. It's also about the fundamental human drive to understand the building blocks of life, to tinker, to experiment, to push the boundaries of what's possible. This movement has the potential to solve some of the most pressing global challenges. Imagine citizen scientists developing sustainable food sources, engineering microbes to clean up pollution, or creating novel solutions to combat climate change. The democratization of scientific knowledge is a key driver. Open-source platforms and online communities are sharing protocols, data, and expertise, fostering a collaborative environment where anyone can contribute. The rapid development of affordable, user-friendly tools is fueling this revolution. What once required expensive equipment and specialized training is now accessible through relatively inexpensive kits and online tutorials. CRISPR technology is the catalyst that has truly ignited the DIY biology movement. This revolutionary gene editing tool allows for precise and targeted modifications to DNA sequences. CRISPR has dramatically lowered the barrier to entry for genetic manipulation. It's simpler, faster, and more affordable than previous gene editing techniques. This accessibility has fueled the surge of biohackers and citizen scientists eager to explore the possibilities of genetic engineering outside traditional institutional settings. They are driven by curiosity, a desire to innovate, and a belief that science should be open to all. However, this newfound power comes with significant risks. The potential for unintended consequences looms large. Gene editing, even with CRISPR's precision, can have off-target effects, leading to unforeseen mutations and unpredictable outcomes. The creation of unforeseen biological hazards is a very real possibility. The lack of regulatory oversight in a DIY environment is a major concern. Without proper training and safety protocols, experiments could inadvertently create harmful organisms or release genetically modified materials into the environment. The potential spread of modified organisms is a particularly worrying scenario. A seemingly harmless experiment in a garage could have devastating impacts on ecosystems and human health if it escapes containment. DIY biology challenges the established scientific order, disrupting the traditional hierarchy of universities, research labs, and government agencies. A decentralized network of amateur scientists is emerging, operating outside the conventional structures of scientific research. This blurring of lines between legitimate research and potentially dangerous experimentation raises serious questions. Who decides what is acceptable? Who is responsible when things go wrong? The question of control over the technology is paramount. Should access to gene editing tools be restricted? If so, how can we balance the need for safety with the desire to foster innovation? And ultimately, who benefits from these advancements? Will the fruits of DIY biology be shared equitably, or will they exacerbate existing inequalities? Navigating these complex challenges requires a delicate balance between fostering creativity and ensuring safety. We must encourage innovation while mitigating the risks associated with DIY biology. Robust regulatory frameworks are essential. These frameworks should be flexible enough to adapt to the rapidly evolving landscape of genetic engineering, but also stringent enough to prevent the misuse of these powerful tools. Ethical guidelines are equally important. The DIY biology community must develop and adhere to a strong code of conduct that prioritizes safety, transparency, and responsible innovation. Public education plays a crucial role. Informed citizens are better equipped to understand the potential benefits and risks of DIY biology and to participate in the ongoing debate about its future. Mitigating the risks associated with DIY biology requires a multifaceted approach. This includes developing safer gene editing technologies, improving containment protocols, and fostering a culture of responsible experimentation. Ultimately, the goal is to harness the transformative potential of DIY biology while minimizing the potential for harm. This requires collaboration between scientists, policymakers, ethicists, and the DIY biology community itself. DIY biology represents a profound shift in our relationship with biology. It demands a thoughtful and proactive approach to ensure that the power to edit life is used responsibly. We are moving towards a future where biology is no longer confined to the laboratory. It is potentially in the hands of anyone, anywhere. This democratization of genetic engineering presents both unprecedented opportunities and unprecedented challenges. How we navigate this new landscape will determine the future of life on Earth. Tell us in the comments and let us know what we should imagine next.